So we're going to go ahead and get started, and I'm going to just do a few introductions, and then turn it over to Allison to introduce the space and the facilities and whatnot. But thank you so much for joining us, everyone. This is great. I'm Josh Benish, and I've been working very closely with Hannah Fisher over there, our events and communications coordinator, and others here at the Water Resources Institute to put on this event, and we're really glad to have you all here today. Our institute brings together researchers and scholars from across campus, enhancing the collective impact of UVM's water research. And today we're kicking off the inaugural event of the Talks and Walks on UVM's Waterside series. This series is made possible by all of our UVM contributors, which includes the Proctor Maple Research Center, UVM Field Labs Core, Natural History Museum, the UVM Natural Areas, the Rubenstein Ecosystem Science Lab, the Field Naturalist Program, the Pringle Herbarium, the Environmental Program, and the Climate Measurements Center of Excellence. So it's really this great collaborative effort across all of these different entities across the university. And we're really grateful to be hosted here today in the beautiful Coach Barn here at Shelburne Farms for this first event. We really thank Allison and the rest of her team for donating the use of this space for us. This is quite a remarkable place, and it's really generous of Shelburne Farms to be able to give us the space here today. I want to turn it over to Allison just for brief introductions of the space and some housekeeping things.
Yeah. Hi, good morning, everyone. My name is Allison Spain. I'm the Event and Farm Functions Coordinator here at Shelburne Farms. So welcome to Shelburne Farms on this beautiful fall morning. And welcome to the Coach Barn, which just reopened mid-September after a year of renovations and many more years prior to the renovations actually taking place. But we're very excited to be back in this new space. We've maintained a lot of the originality of this building, but now we have great things like lights and heat, and we have a brand new kitchen and more than one bathroom and a new geothermal system. So we're just really thrilled to be in this beautiful space on this property, and very happy to be welcoming this group, especially for the inaugural event in the UVM Talks and Walks on the Waterside. If you have any questions about the space or about Shelburne Farms, I will be floating around today, and I'm happy to chat. Just a quick note, restrooms are kind of through the east hall over here, around to the left, and then take a right. But you're welcome to explore the space while you're here, and I hope you enjoy your event today. Thanks so much.
Thank you so much.
Yeah.
So the research that we're going to be highlighting throughout this series occurs across our living laboratories, like our natural areas, Shelburne Farms, and Lake Champlain. These places are hubs for impactful research and student learning. For example, as a student, I was able to complete a service learning project here at Shelburne Farms focused on water quality monitoring, which really helped shape the direction of my career. And so I'm really grateful for the partnerships between UVM and Shelburne Farms that provide essential opportunities for both our students and our researchers. And we believe that global change begins in places like this, where we can conduct research, work together, and learn how to be able to make the world a better place. These sites are blueprints for effective stewardship and for translating science into action around the globe. So with this kind of focus in mind, we're ready to dive deep into the global ripples of our local Lake Champlain research, and I'll turn it over to Hannah for a quick introduction for today's speaker.
All right, this is the last introduction, I swear. So I'm going to introduce Jason today. Jason works at UVM currently as the director of the Rubenstein Lab, and he's also a professor. Prior to this, he wanted me to note that he's a really nice guy, and he also worked in a bunch of NGOs and as a biologist on the state and federal level. So with that, I'm going to turn it over to Jason.
Thank you. I got one right here. Yeah, good. Okay. All right. Well, good morning, everyone. Thank you so much for taking time out of your weekend, especially on what's turning out to be a really beautiful day. I told my wife this morning, I said, I hope it stays cloudy, maybe rains a little bit. We'll get more people coming in because it turns out good. But anyways, thank you for taking your time out, and thank you to Beverly for inviting me, and to Josh and Hannah for setting this up. I'm gonna, as the title says, I'm gonna talk a bit about, I'm gonna talk a lot about how the research we're doing at the Rubenstein Lab goes well beyond Lake Champlain. So when we have people come to the lab, I often say, yes, we work on the watershed, we work on the lake, but we work on so much more beyond Lake Champlain and beyond Vermont, beyond New York. So hopefully I'll give you a flavor of this today. There we go. Before I start, I just want to acknowledge all the things I'm going to be talking about today cost money and time. And so these are all the different agencies, institutions that have contributed to the work I'm going to show today. I'm not going to show all the work, and it's not all the institutes that fund us, but I really want to a shout out especially to Senator Leahy and the Great Lakes Fishery Commission. Many in the room have been touched by Senator Leahy's efforts to bring funding and effort into the Lake Champlain and protecting it. So we are no exception. We have received a lot of federal fundings and continue to, even though the senator has retired, his legacy for continuing to support Lake Champlain carries on. And the Great Lakes Fishery Commission, working with Senator Leahy, was very instrumental in much of this work, much of the research we're doing, and also for the new research vessel we have, which I'll talk a little bit about. So just wanted to give a shout out to everyone here. A disclaimer that this is going to be very focused on the Rubenstein Lab, and in particular focused on fish and zooplankton. We do a lot more at the lab than fish and zooplankton, so we do watershed science, hydrology, limnology, which is part of zooplankton. We have a great education program through the Sea Grant program, Watershed Alliance program, and of course we do fish. And it's a large community. I counted up. This is our board of everyone. We have 68 people at the lab, ranging from undergraduates all up to faculty and everyone in between. When I got here in 2011, there were about 15 people working there, so we've really grown the program. And we rely on each other quite a bit because we're out in the field, and there's always danger when you're working in and on water, but also in the lab, there's a lot of stuff that can go wrong. So it's a really tight-knit community. Even though we're diverse in what we're studying, we are really tight-knit. So it's a great place to work. And again, just wanted to disclaim, if Mindy Morales, our limnologist, was presenting, she could tell you a whole new direction of international research that's going on. And Jefferson, the same thing. So I just want to, this is not, what we do is not limited to fish and zooplankton, but that's what I'm going to show today. So one of the things I hope you take home today is that Lake Champlain is really, really interesting and unique, not just because we live next to it, but there's some features of it that are really make it special in the world. And it's due to a combination of things. And the first of all is the watershed to lake ratio. I'm sure lots of you have already heard of this. What happens in the watershed affects the lake because it's just such a massive landmass compared to the lake size. And I just put some other, Lake Baikal is also pretty big, 17-1. Lake Tanganyika in Africa is 9-1, and Lake Superior in the Great Lakes is less than 2-1. So we're somewhat unique in that we have a large influence of the watershed on the lake. We also have a highly fragmented lake. So we have a lot of islands naturally, but we have a lot of human-made connections between the islands and the mainland that bisects the lake into different basins. And so this is the 5-kilometer causeway between Mallets Bay and the main lake. And there's only 2 small openings along those 5 kilometers to let boats through. And so if you were to go back in history, this wouldn't be here, so there'd be a stronger connection between Mallets Bay and lake. The same thing, sandbars between Mallets Bay and the northeast arm. Missisquoi Bay is somewhat cut off from the northeast arm, and then South Lake is kind of naturally constricted compared to the open lake and very shallow. So it's highly fragmented. It's a large watershed to lake area ratio. Somewhat because of that fragmentation and because of the different basin morphometries, we have varying trophic states or productivities in each of those basins. So in a way, we almost have 5 lakes in one lake in here. So we have Missisquoi Bay and South Lake here. They're very high productivity, so very green, as you know, cyanobacteria blooms. Northeast Arm is also very productive and somewhat deeper And then we have the main lake, which is less productive, and then Mallet's Bay is the least productive out of all the systems. But we have this gradient, and I'm going to keep coming back to this idea of gradients. We have representation of all different types of productivity across the lake in one lake. And then we also have, because of that, we have various communities. I put fish up here, but there's different plankton communities, there's different invertebrate communities. And so we're going from these fish that require cold and deep water, which we find in the main lake, all the way to these fishes that are more warm water and can maybe tolerate less oxygen, and everything in between. So we've got lake trout, smelt, we got walleye and whitefish, perch, the basses, white perch, alewife, and invasive tench, and so forth. But these communities all vary across the different basins. So we're really set up well to really think of Lake Champlain as an experiment, as an experimental gradient on which to learn things. And so here visually, you see the different bays or different basins with different levels of productivity. We can line those up from least productive to most productive. And we also have different shapes and morphometries of the basin. So we have the main lake, which is the deepest, all the way to the south lake, which is shallow. So we have these gradients of conditions across the lake. And of course, as I just mentioned, we have different communities. So we can think of this as kind of an experimental setup. And I'm gonna So we can think of it as an experimental setup on which we can kind of do experiments and see how the systems respond as a function of these different gradients. And just as a quick example, in science, we know that systems that are more diverse are less vulnerable or less susceptible to invasive species. So if you have a lot of species there, it's hard for invasive species to kind of get a hold. But we also know that this relationship is also, the productivity of the system also is very important. So when you have high productivity, there's more food available in the big picture, so there's more room for invasive species maybe to get a foothold, whereas in systems that have very low productivity, there's not a lot of food in the bigger sense, so it might be harder. And so we still have this relationship with diversity, but overall, the system productivity may have a positive effect on vulnerability, even though diversity may be decreasing it as diversity goes down. So just trying to loosen up your brains, thinking in terms of science and experiments. And then on top of that, there's other things that are important. So does the system have a lot of predators already or not? And if we consider that, if we have a system that has very few predators in it, then we might have more vulnerability or susceptibility to invasive species. And if we have a system with a lot of predators already in it, maybe that lowers its susceptibility. And two great examples, the Great Lakes, back in the 1900s, invasive alewife came in. The lake trout population had already been crashed because of overfishing, sea lamprey, habitat loss. And so those systems had very low predation vulnerability. So wherever you are on the spectrum, it may have jacked up their susceptibility to alewife or smelt coming in and made them more susceptible. Here in Lake Champlain, we have a really good stocking program for half a century. The fish are doing well, so we had a lot of fish. And so when the invasive alewife came in the mid-2000s, we were probably lowering the impact of invasive species of alewife because we had a bunch of predators sitting there waiting to eat them. So you can see how we can use Lake Champlain to start asking questions like this. And so I'm going to go through some examples here. One example of looking at how the system might be influenced by the gradients that we have. So alewife came into the system in around 2003. They were first discovered. They didn't really take a foothold until 2007. And if we look at the abundances of the invasive alewife from 2007, they're there, they're going down over time. And we also have the density of alewife, or of smelt, native smelt. And you can see over time, they were doing fine, and then even when alewife came in, they stayed pretty high up. So no negative effect that we can tell of alewife on smelt in the main lake. But if we look at Mallett's Bay and the northeast arm, well, let's look at Mallett's Bay first. What we can see is we have alewife in there, but we see a drop in the rainbow smelt densities or abundances since the alewife came in. And if we look at northeast arm, the same picture has it. They're there in the northeast arm. There's no trends, but we see this decline in rainbow smelt. So what could be causing this? If we look at the predators in the system, both Mallett's Bay and northeast arm, they have very few cold water predators. So there's very few lake trout in those parts of the lake because it's too warm. It's hard for them to get back and forth between the systems because in the summertime the water's warm. They have to go shallow to get in, and there's none there. At the same time, we didn't see any change in the plankton or the prey of rainbow smelt in these systems. So they're not going down because of predators. They're not going down because of food. But what they are going down is related to the environmental conditions. So in the main lake, smelt like cold water, and they need oxygen just like we all do. And so this is the habitat availability for smelt in the main lake across the different months over years. And so the blue bar indicates the amount of depth in the habitat that is suitable for the smelt. And so you can see as the summer progresses, the surface water is warm, and that's not good for the smelt, so they stay deeper. Okay, so there's still plenty of habitat available for them, and then it cools down in October. But if we look at the other two basins, what we see is they have the same effect. The surface waters get warm, so the habitat goes down. But you can see below, they're getting squeezed from below because those systems are going anoxic, so the oxygen is too low for them. And so they're being squeezed from the top by temperature, and they're being squeezed by lack of oxygen from below. And these combinations will squeeze the population into a very small space where they may be competing with each other. There may be increased risk of disease transmission, and there may be more competition with alewife, which are in those systems. And so we think that the environmental conditions of the lake across these gradients are affecting the interaction between smelt and alewife. Okay, so again, think of that experimental gradient. Now, so that's an example. And so the other thing about Lake Champlain is, it's a big lake. You look out there and it's a big lake, but it's not really big compared to the Great Lakes. And so you can kind of see the scale here. But the system in Lake Champlain, the communities are very similar to the Great Lakes communities, all of the Great Lakes communities, because we have that gradient of conditions. We got warm, productive waters, Lake Erie, and we've got deeper waters, which are colder and maybe less productive, like Lake Michigan or Lake Superior. And so we kind of been pitching this as a mesocosm of the Great Lakes. And so we have that comparative aspect going for us. We also, because of its size relative to these large lakes, Lake Champlain is much more tractable.
Jason, when you talk about productivity, are you talking about total biomass?
I'm talking about total biomass, total like the plant life, phytoplankton, transferring all the way up to the fishes. So overall system productivity. So because of its size, it's big, but it's not Lake Superior big. We can learn things from it because we can know what's going on without as much effort. And so as an example of this, some of you may have seen, we do acoustic telemetry. So we can plant these little tags inside lake trout or walleye or sturgeon, and they emit signals every minute or two. And we have these listening devices throughout the lake. And if a fish swims close enough, they start talking to each other. Like, hey, I'm here. Okay, who are you? Here's my tag number. Okay, we know what fish you are. And when they swim by, they talk, and so we have a record of the fish moving there. And what we can do is put out receivers. And here's just an example. Here are all the receivers in Lake Champlain, and you can see it's covering pretty much most of the lake. Lake Superior, because it's so big, they're only getting kind of the embayments and along different shorelines, but the coverage of the lake is lacking just because of the size. Here we can tell where the lake trout is going from different spawning ground to different spawning ground within the spawning season. Lake Superior, they could be going anywhere. So this is what I mean by its attractable size. So that makes it attractive. And then the final thing is we're easier and cheaper to access than the Great Lakes. We have all the capabilities of the large research vessels on the Great Lakes. So we're fully capable, but yet we're a quarter of the price to rent out, if you will. And we can get access to most of the lake easily within a couple hours. And so as one example, as Hannah was saying, I used to work for USGS here in Chequamegon Bay in Ashland, and I ran a station for the USGS. We had a 100-foot research vessel. It would take us 2 hours to get to 100 meters depth. Here in Burlington, it takes us 15 or 20 minutes. So it's an attractive place. We pitch this to our colleagues in the Great Lakes, and some of them come and do work here to test ideas out to see if it would work on a bigger scale. Okay, so where are we going to go next? So what I'm going to do, now that you've kind of got the context of Lake Champlain, I'm going to talk about some of the projects we're doing. I'm going to start with fish projects, and then we're going to go on to zooplankton, and then we'll finish up. So hopefully you can see some of these ideas in action. So the first one is lake trout restoration. I'm sure you've all heard it's been a great success story here in Lake Champlain. So in the Great Lakes, it's been a different story. It's been a long effort. It's been lake trout restoration has been going on for more than 100 years. And aside from Lake Superior, it hasn't been very successful. And here's an example. Lake Superior has 4 different types of lake trout. This is called the lean lake trout. This is what we have here in Lake Champlain. And then we got 3 other ecotypes. This big bruiser is a deep water fish. It can be up to 75% fat. It's all lipids. So very different lifestyle than the lean lake trout. And then we've got these other forms. So Lake Superior is good. It wasn't as impacted by commercial fishing as much, and it had big space, so it was kind of a bit resilient to overfishing. Some of the impediments that historically were happening in the Great Lakes and still today is sea lamprey predation. That's a big issue. Negative impacts of alewife, which I'll talk a little bit more. Alewife will feed on young lake trout, so that's a negative effect. But also, alewife is full of thiaminase, which is an enzyme that breaks down thymine, vitamin B. And if the predators eat too much of that, it affects their reproductive organs, and they're not as successful. So that's kind of a double whammy for alewife. Habitat loss, and then historically, overfishing really decimated lake trout. It's been a hard time getting the Great Lakes to recover from that. We have had the same issues in Lake Champlain. They were gone by 1900, not really sure why. And despite stocking efforts by the states since 1973, they were reproducing, but the young fish were hatching out, but they weren't surviving. So for 50 years or so, nothing was happening. And again, we thought this was because of really high sea lamprey wounding rates, much higher than the Great Lakes. And also, especially when the alewife came in 2003, management thought, this is just, it's really bad. It's a really bad situation. But it's been a success story. And we're going to talk a little bit about that now. So in 2015, Ellen Marsden discovered wild lake trout, first time ever. And how do we know? I put a little note here to remind me. How do we know a fish is wild? Every fish that is stocked into Lake Champlain gets a fin clip. So they have 5 fins. They have 2 pectoral, 2 pelvic, and an adipose. And every year, all the fish that are put in the lake get one of the fins clipped. So last year might have been the left pectoral, so every fish. The next year, it's the right pectoral. The next year, it's the pelvic. And so if you catch a lake trout in the lake and it's missing a fin, it was stocked. And depending which fin it is, how big the fish is, you say, oh, that one was stocked in 2018, this one was stocked in 2019. So we know, but what Ellen found was that she started finding fish with no fin clips in really high proportions starting in 2015. And so you can see over the years that the proportion has remained around 50% plus or minus. It's still going today. So we have all these wild fish hatching out and surviving. They're surviving to adulthood. So the state does a gillnet survey every summer, and they're looking at the ratio of adult fish that have fin clips and don't have fin clips. And you can see that that proportion of wild fish is increasing and it continues to increase. So this is great for restoration. So now we have more than 13 year classes in the lake that have been produced that are wild fish. And we're going through time now that there should be at least 7 different year classes that are adult mature spawning. So it's just going to keep going. This is great. If you're a hatchery, you want to put yourself out of business, and that's what happened here. So the states decided to stop stocking entirely lake trout in 2025. It's good. It's a success story. But the question is, well, why? Because we have invasive alewife came in, and we have really high sea lamprey wounding rates. And same conditions as the Great Lakes, but yet it's working here and not there. So it's unexpected and unparalleled. And we tried to investigate some of this with some modeling. And what we found was that it seems like the alewife fueled the restoration as opposed to inhibited restoration. And so just some quick, so we were interested, we're going to use this, we used the model to evaluate how alewife may have affected the food web and lake trout. And so, as I just said, they appeared in the mid-2000s. Lake trout were being stocked since 1973, and the first evidence of wild fish was the 2012 cohort that we found in 2015. And so we did this model, food web model, who's eating who, how much. And what we found from the model was that the other prey species, in addition to alewife, so the rainbow smelt, the trout perch, they didn't really change in the main lake. In the other part of the basins, they said the smelts are changing, but in the main lake, there was no change. The model predicted that the lake trout should switch to a diet of alewife, which you can see kind of here in this figure, going from nothing to very high. Smelt decreased in the diet, and that in the model, the biomass, the amount of lake trout grew because of this influx of alewife. In the real world, we did see lake trout switch to a high diet of alewife. They actually did that. We didn't see really any drops in the main lake with other species. And in the model, it was the stocked lake trout that increased biomass, but what we saw is the wild fish blew up. So the model is showing what we're seeing in the lake. And so what this is suggesting is that alewife were good for lake trout restoration in Lake Champlain, that there wasn't really a negative effect of alewife on the other community members. It seemed to have jump-started the recruitment of lake trout, and that maybe there was some unoccupied niche in Lake Champlain that the alewife fed and have a more direct line from the bottom of the food web to the lake trout. I don't know. I don't think anyone knows. There are various theories of angler bringing buckets of alewife into fish with. So it's. I think they were discovered in Missisquoi or Northeast Arm, which is not the nearest point of entry if they were to come up like the Richelieu River or through the Hudson. So. I believe so, yes.
Why are the wild lake trout in Lake Champlain not affected by the diamond?
That's a good question. We have done some work, we meaning Ellen Marsden. So the thiaminase problem has been found in hatcheries. And no one has actually looked at the problem in the lake. And so what Ellen did was she had some eggs that she raised in hatchery environment, and she put some eggs out in the environment and protected. And it seems like the eggs and the embryos in the little fish can absorb vitamin B from the environment. There's a lot of vitamin B just in the environment, and so they can absorb. So the thiaminase problem, for some reason, is not an issue here in Lake Champlain. I don't know if they've done those work in the Great Lakes. So Again, this is all unexpected. We don't know why. Really, we don't know why, but we think alewife jump-started them for some reason. Okay, so where are we going? We're going to go. Okay, so Lake Champlain is ground zero for understanding evolution and diversification. This is a big sentence. Okay, evolution right here, ground zero, Lake Champlain. So it's a little dramatic, but you'll see why I'm saying this. So I'm going to start off with Lake Superior again. So Lake Superior is 10,000 years old, glaciers, and we have 4 different ecotypes. So this is diversification of lake trout species, and they all occupy different habitats, so kind of separating niches. Before commercial fishing, there were up to 10 different lake trout types. So this is kind of narrowed down, but it still exemplifies what can happen over time in large lakes. Here in Lake Champlain, we have this opportunity that the fish have been stopped. We know the strains of lake trout that were in the hatcheries that were thrown in the lake. And now we have this first generation of fish being produced in the wild under natural conditions. And so the opportunity is, well, let's assess these fish, let's take pictures of them, and then we can start looking at their phenotypic variation, so how their body shape is, and is it changing, and is it different between a stocked fish and a wild fish? And so this is basically the start. We're taking a new species, putting it in a system, and we can watch it evolve, so to speak. And the first step in that is looking at variation in its physical characteristics. And we can use that and compare it to 10,000 years of diversification and evolution in Lake Superior. Okay, so great, ground zero of evolution. Part of evolution is also genetics, and so we, and by we, I mean other people who are geneticists, not me at the lab, so Thais Beranos. And I should mention that Peter Euclid mentioned here, he's at Indiana University. He's leading this charge and working with our faculty, Thais Beranos, who's our new bishop. I got to remember to start acknowledging people because this is not all my work by any means. So we have this team working on this kind of phenotypic diversity, but they're also taking genetic samples of all these fish, and we have genetic samples from other lake systems. And so kind of this ground zero of evolution, again, we have Lake Champlain, hatchery fish strains. We're getting the first generations that are reaching adulthood and spawning, but we also have fish from these other lakes. So as I said, Lake Superior is 10,000 years old. We've got this diversity here. We have Lake Mistassini in Quebec, just a couple hundred kilometers north. Lake is somewhat similar in size to Lake Champlain, and it's also got this diversity of fish. This lake has never been commercially fished. Lake Superior has. Our fish are not commercially fished. So we're starting, again, seeing a gradient over time in different systems of ways to learn more about what may happen here in Lake Champlain. Now, on top of this, I should also mention that Thais Beranos, our fisheries faculty member, this is her work here in Lake Mistassini. So she's involved in this. And we also have an opportunity with Lake Ohrid in North Macedonia, Albania, which is in the Balkans. This lake is millions of years old. It's also got trout species. It's got 2 endemic species, the Velvicha and the Ohrid trout. And these trout also have these different, maybe not as much phenotypic diversity, but they have behavioral diversity that they're spawning at different times of year in different environments. And so we're just starting a project with biologists here in North Macedonia, and we can look across this gradient of time in lakes and diversity to start understanding how species diversify and evolve. So it's very exciting. Also going to Lake Ohrid. So Lake Ohrid and Lake Champlain are sister lakes. So there was a formal establishment of this in the 1990s. They did a lot of work together, watershed work, and that kind of petered out in the mid-2000s, 2010. And we just got, just in the last year, we got requested from the US Embassy there to start reviving this and establishing collaborations. And so I was over there in September, and we're starting a fisheries conservation management project on the Lake Ohrid trout. So Ohrid trout is endemic. It's emblematic of the region. and it's a major commercial fishery, and it's also very culturally important. And so they, their fish stocks have been going down. They have a big hatchery program for 90 years, but they've never evaluated their hatchery program to see if it's successful or not. So the question is, when they're catching the fish in the wild, is it because of the effort in the hatcheries, or is it because wild fish are producing enough and the hatcheries are not contributing anything? So we're working with them to evaluate what proportion of the commercial fishery is hatchery versus wild produce. And this is just a quick schematic. We're going to start this January when the fish are spawning. They capture the fish, they take the eggs and milk, mix them up, and then put them in a hatchery, release the fish. We're going to be taking genetic samples of the adult spawners they catch and start doing genetic mapping of their genetics. We're going to take genetic samples of the fish they're raising in the hatchery, put that all also into this genetic mapping. These fish are released into the lake. There's some subcomponent of the population that is also probably reproducing successfully in the wild, so those fish are also contributing to the population. These fish will grow up. When they're caught commercially, we'll take genetic samples, and then we will map what we find in the commercial fish back to this database to the commercially caught fish, and basically be able to understand their parentage. So did it come from a hatchery using this technique? Did it come from which hatchery did it come from? And if it didn't come from any hatchery, we infer that it's a wild fish. So this will give them an idea of, is the commercial fishery being supported by hatcheries or not, or some blend, and then that will lead into their management decisions. This is something we've done here, and we, again, I don't mean me, I mean the Fish and Wildlife Service and Thais and others. We've used this parentage-based tagging genetics approach in Lake Champlain for Atlantic salmon, and it's also been used elsewhere in the world to identify different populations of salmon and other species. So it's a well-proven method, and we're fortunate to be able to work with the Macedonians and the Albanians on this project. Okay, so switching gears a little bit, so we've got this new boat, which hopefully you've all seen in one form or another. It's a hybrid boat, so it's electric. So think of a Prius, electric and also diesel. It's big, it can hold a lot of students, so it's been really good for us. So one of our students, Mia McReynolds, she's doing her PhD on global fisheries assessments. And so in the world at large, a lot of fish stock assessments are taking off of research vessels using sonar, so basically scientific grade fish finders. Using sound, they can look at the reflection of sound and basically estimate how many and how much fish are in the water column below. So this is used in the Great Lakes, it's used in other lakes, it's used in the oceans. But the thing is near the surface, it's not a lot of volume samples, so we don't really have a good metric near the surface. And we also don't know what the boat's effect on the fish behavior is. And so with our new vessel, well, before that, we procured some of this really cutting edge acoustic equipment where we're putting it on the bottom looking up. And it's got batteries, it's saving all the data. And so it's looking up in reverse of this so we can see close to the surface. So we can see what's going on there. With our new vessel, we can run in silent mode. So again, think of an electric car, it's very quiet. So we can go over the water that, and using these, we're driving over these transducers and looking at the fish distribution near the surface. And then we'll go back over and use our diesel and again compare, are we seeing no fish or less fish compared to when we're running it over electric? And we've been doing this experiment for 2 years now, many times, and this is forming Mia's dissertation. And this, again, kind of global, this is something that all countries that are doing acoustic assessments in the ocean and in lakes are going to be really interested in this, because if there is this effect, that means everyone's assessments are going to be biased low. So it's really fascinating and exciting. Okay, so now let's go on to zooplankton. And so before I got to introduce you to GLEON, the Global Lake Ecological Observatory Network. So a lot of our zooplankton work is within this organization. And I'm going to explain a little bit about it because it's really unique. So 3 words to describe it: networked lake science. Their whole goal is to bring people together, to share data, to ask questions, and to evaluate these questions across broad lakes. Not just asking a question for me in Shelburne Pond, but asking my question for Shelburne Pond and then 20 other lakes around the world, which may be similar to Shelburne Pond or may be a gradient of lake types. And so this is just, it's a grassroots organization, and they're basing it off of this, this is a term I just learned, socio-technological approach, where humans are integrated into the information system, using, reusing, generating new data. And so you can see this life cycle, new members who are producing data, reusing data, making sure things are reproducible with data and models, and generating information. So I have to, I have to thank Beverly for this whole aspect of this research, because I arrived here in 2011, I met Beverly, and in 2013, I was talking, she goes, I was on sabbatical at the Carey Ecosystem Institute in New York, and someone there asked me to go to this lake meeting, and I'm a watershed person, and this person wanted to bring watershed into the lake, and it's like, I don't really want to go, or really want to go alone. Will you go with me? And so we schemed up and found some funding, and so we ended up going together to our first GLEON meeting, which was in Argentina, and it really, it really set the track for a big part of my research in the lab's research, as you'll see. So this was great. And so within GLEON, again, just a broad view, there's different ways of doing science, and within GLEON, they kind of embrace all these different ways: natural history, experiments, theory, comparative studies, and they're really focused on working with people, taking advantage, harnessing people's knowledge of different ways of looking at things, how they communicate, what they bring to the table. They're also very engaged with data sharing, giving credit, and also, of course, the lakes, and looking at how do we look at lakes long-term, consistently, and put all these things together to come up with good research that talks about roles and responses of lakes to climate change. And so all these things are happening in GLEON. It's a really wonderful organization. And the way it works is different. So if you think of going to a science conference for a week, you sit there and you listen to talk after talk, and you may get bored after a while. You may feel like that. It's kind of like this. I'm just talking at you here. There's not a lot of interaction, so hopefully you're not being bored. But think of doing this for a full day, for 5 days. The engagement, the interaction is not the best. But at GLEON, the whole idea is to get people in rooms and start talking about ideas, to share ideas, and then hopefully launch projects. And in this meeting here, over half the people at that time were students, were graduate students or undergraduate students. So it's a great way of getting science done. And so where are we going? So since 2013, the whole Rubenstein Lab has been engaged in many different ways in GLEON, and it provides so many benefits. First of all, we get to network with people across the whole world. We're all developing leadership skills and learning to apply them, whether we're a student or whether we're someone like me. There's a lot of big data in there, so we're developing those skills. Anna, who you'll meet later on the walk, she's heavily involved. You can see some pictures of her here. She's a leader in the Student Association. So she's developing people and project management skills. If you're on a project with 50 people from around the world, you're going to get people who are at your career stage, and you're going to get people below and above. So if you're on a project, how does Anna handle someone like me who's at the end of their career and maybe a bit, power dynamic stuff going on? We learn these things in these groups and how to handle situations and how to manage people and projects. So it's a great learning ground. And we have poster sessions. So here's Sadie, she was an undergraduate, and John was a postdoc. So it's just a really great organization to get involved in. And so what I'm going to do is just quickly talk through one project that's currently ongoing now. We call it ZIG, Zooplankton as Indicators Group. Zooplankton are the microscopic rice or smaller size animals that feed on phytoplankton, and they're an energy conduit between the base of the food web, the phytoplankton, the plants, and fish. So they're an important part of the food web. So in this project, it formed out of Gleon in 2019. We have over 100 people engaged in it. And basically, we have time series of zooplankton and other limnological variables from over 300 lakes around the world, and you can see them all here. And we've just partitioned this into different thermal regions of the world, which you'll see this in the next couple slides. Huge effort. It's taken 5 years to put all this together, to QA, QC it, and now it's been submitted, it's in review, and hopefully by next year it'll be published and it'll be accessible to anyone who wants to look at it. So in this project, we can do simple things like, let's look at the diversity in terms of richness, the number of genera in each lake over latitudinal gradients. And what we can see here is that although the sample sizes are different, on average, there's more richness in the northern latitudes than lower latitudes. So that's pretty easy to do. But we can also ask more functional questions. And so this is about as technical as I'm going to get, I think. So something called resource use efficiency. It's a measure of function in an ecosystem that has implications for water quality, for energy flow through the system, how much fish can be supported ultimately. So it's not just like how many species are there, it's like how is the system functioning. And so this is simply the ratio of predator biomass to prey biomass, so getting at your question. So if we have this much phytoplankton biomass in a system and this much zooplankton biomass, we can just simply do a division and get some sort of percent, like 10% is being utilized at the next trophic level. In a system with lower zooplankton and the same amount of phytoplankton, this would have a lower RUE. So I'm going to show a couple slides showing patterns of RUE as a function of other parameters. And we'll circle back eventually to Lake Champlain on this. So if we just look at this metric of trophic efficiency across these different thermal regions, which is really latitudes, you can see the RU seems to be a bit higher in northern latitudes and kind of goes down as you lower in latitude into the southern hemisphere. If we look at these values of RU against surface temperature of lakes, we see there's variation. It seems to go down at warmer temperatures and maybe at colder temperatures, but there's not really that strong of a relationship there. Again, we're looking at ecosystem function across these large global gradients. But if we look at RU as a function, RUE, the resource use efficiency across, this is total phosphorus. So this is a proxy for the productivity of the lake. So lakes that are highly productive are at this end, lakes that are clear and very low productive are at this end. And what we can see is that again, there's scatter, but in general, the efficiency declines as the trophic state of the lake increases. So it's if you have more there, you're not utilizing proportionally as much as you can. So again, you can see how this varies across latitudes, but overall, this is a pattern. And then finally, if we look at this measure as a function of zooplankton community composition, so copepods are a type of zooplankton. You may know Daphnia, the water flea. All the experiments are done with it. So it seems like there's a strong relationship, again, relatively speaking, between the resource use efficiency in lake systems and how many calanoid copepods is in the community. So these are all new things we're seeing because this data set is so extensive. It's the most spatially and temporally extensive data set in the world. So this is a first look at it. So how does this relate to Lake Champlain? Well, you'll remember that we have gradient of productivity in Lake Champlain. We also have different depths and morphometries and different fish communities. And so we can use Lake Champlain in this global context And we can do it because we're fortunate to have the long-term monitoring program on the lake. So this has been going on, I think the water quality has been going on since 1992, the zooplankton since 1995, and we have access to these data. And so we can start, we can contribute these data to this global data set. We have 15 sites across all the different parts of the lake since 1995 at relatively high frequency. And so we can contribute to these global questions, but we can also ask the same questions within Lake Champlain to see what we can learn. How well can we use the experimental grade Lake Champlain to ask questions at a global scale? So it's kind of both ways. And also, as you know, we've had lake trout restoration, we've had alewife invasion, we've also had invasion by these spiny water fleas and other zooplankton. So because we have this data set across all the basins and we have the environmental conditions, we can start asking questions like, oh, did the alewife invasion affect the food web community equally across the basins, which we talked about already? But with the lake trout restoration going on, we have a lot more lake trout in there. Are they going to eat up all the fish, which then releases the zooplankton from predation? So we might see a response at that level, which may clear up the lake better because the zooplankton can eat a lot of the phytoplankton. So these are all questions we can ask with this wonderful data set across the 5 lakes in one of Lake Champlain. And further, we have other invasive species knocking at the doorstep. We've got quagga mussels, which have done a real number in the Great Lakes, and also round goby. So we can start, again, now we can start assessing relationships and function across these gradients to see which parts of Lake Champlain may be more or less susceptible to quagga mussels, to round gobies. Is there anything we can do to prepare for that? And when and if they get here, we can evaluate that through time because we have these time series of monitoring data. So, okay, so that is, that's a Gleon project. Anna did not know I was going to show her research up here. So we also have students like Anna and others who are doing other international projects, not necessarily through Gleon, but because of networks through Gleon and our global connections. So Anna is looking at carbon flux. And in the oceans are a big carbon sink. So carbon is taken into the and fixed by phytoplankton. They're used in the food web, but they also die and they sink down. And so there's a flux that slowly goes down over time. But we also have zooplankton that eat the carbon, and they undergo migration up and down every day. And so when they eat, they go down, they poop, and that's just transporting concentrated carbon much quicker because it's active migration versus things that are just kind of settling down. Big research topic in the oceans, nothing done in lakes. So Anna's work is looking at the lake component of this question, and she got connected with our colleagues in Germany who have this huge research facility. Each of these circles are 30 feet in diameter and 60 feet in depth, and you can manipulate them. So add fish to some, add nutrients to the others, and then observe what happens. So experiment. So Anna has been worked over there for a year and is doing these experiments to evaluate this carbon flux migration as a function of fish and zooplankton behavior. So great experience. We have other students who are working in the Netherlands. We have other students who are working in France. So it's, again, kind of a global connection. So my last thing is, this is more or less hard science or science active. This is more about how we do science. So this is, so team science is really kind of blown up in the world. So historically, you might think of this hierarchical structure of science. You know, I have my lab, the students come in, they work under me, I control everything they do. But team science is more like the Gleon model of like, we're working in this together. So there's a lot of papers, a lot of work done on how do you work as a team? How do you decide what you're going to do with a bunch of people? What happens when there's conflict? How do you decide who's the first author? Who's doing what work? What approach are you going to use? So there's a lot of that out there. But one thing there's not a lot like, well, how do you write as a team? And so historically, typically one person would write it and hand it to everyone else, and they would give their comments, and the person does it. Or people would say, you write this section, I write this section, and we're going to staple them together. So this kind of came to a head for me back in 2018. I was working on this paper with 50 people from all over the world, and they were all giving me feedback, and I couldn't keep track of it, and some of it was really good, and some of it was like vague. I had to interpret it, and so I got really frustrated. And so I just said, I need a paragraph on this subject. And I sent it to someone and said, can you just spend 10 minutes and just write everything you know about the subject on? And he did, and he sent it back to me, and I'm like, oh. So I edit it, I revise it, and I'm like, oh, let me send it to her and say, can you spend 10 minutes on this? Easy for anyone to do that. I got it back, I revised it, and I sent it to the third person, and then I had a paragraph, a really nice paragraph that was far better than anything I could do myself in much quicker time. And so this is what I'm calling the we write model. And this is kind of the flow pattern. And we're actually doing a study on this right now. We have groups from all over the world who are writing papers either using a traditional writing model or using this model. And we're doing surveys before and after they do this. And we're going to see if this writing model is helpful or an improvement over writing alone or Frankensteining things together. So that's the last thing I have to say. Just take home messages. I hope these are the things I was thinking, like, what do I want people to remember? The one thing is that our research at the Rubenstein Lab, and this goes for probably all across campus, is that the work we do is not just on Lake Champlain because we have a big boat. It's far broader than that. And that also we should be feel very lucky, very fortunate, very proud that we have this really unique system in the world of experimental gradient, the right size to scale up or down, and to ask these different questions that are broadly applicable across the world. It's really really wonderful. And then, as I mentioned, our students are getting all these international experiences, which they're going to take with them. They're going to be confident whether they're going to a Vermont biologist position or whether they go overseas and become a scientist in a German lab or wherever. They're having these connections. And then finally, I haven't said anything about this, but maybe once I say it, it's all these international collaborations are ways of doing soft diplomacy, and this is, I think, really important. When I was in France last year during the elections and got the election results, people were coming up to me, and I was who I am. I wasn't some stereotype on the board, and it was really important, I think. And I was just in North Macedonia. The ambassador said, we no longer have USAID. I need to figure ways out to engage with the North Macedonians. Can you engage with science? I said, sure. And so I had a wonderful week meeting all different businesses, NGOs, scientists, politicians. And it was really a way of kind of doing this soft diplomacy of collaborating, given the federal landscape as it is for kind of internationalism, nationalism. So I think that's also a very important point, and something I wanted to make in all this. So anyways, thank you very much for listening, and happy to try to answer any questions. Yeah. Should I talk to him? Yeah, great.
Yeah, in Lake Champlain, I'm thinking about lakes and ponds in Vermont and the algal blooms, cyanobacteria. And are there studies, here or like in the simulation tanks in Germany that relate predation of, for instance, trout on the zooplankton, and then the zooplankton decline, and so you get blooms?
Yeah, there's a lot of that work going on, and that's kind of it's kind of like they call it a trophic cascade. If you change one trophic level, like fish, by stocking or removing, do you see a response in the next lower level, and then do you see a response in that next lower level? So they call it biomanipulation in the management world. It works in some cases. In other cases, it doesn't.
Have we seen evidence of that in Lake Champlain?
No, but that's what I'm getting. We're going to be able to ask that question, especially with the lake trout restoration that I allude to. Like, that's a wonderful, because the lake trout are really abundant now, and they weren't, and we have all that data. Yeah. Yeah. But we also have, if I were a betting man, when quaggas come in, I think we're going to see drastic improvement in water quality, but it may be at the expense of fish, because they're going to suck out all that pelagic nutrients and productivity, and that's going to affect the food levels up.
So, Jason, it sounds to me like it's going to be endless manipulation. Do you think about that? I mean, this phenomenon happens because the wild trout came, and then the zooplankton phenomenon happened, and so then there's a new species.
Something happens, or invasive species come.
But you have enough data to say, oh, let's try that.
Yeah. Well, I think management agencies are maybe hesitant to say, oh, we've got quagga mussels, and so let's bring in round gobies because round gobies eat quagga mussels, but round gobies outcompete other things. So I if we were 50 years ago, I think yes, we would be doing that. But there's a book out from someone named Stuart Pym maybe 30 years ago called The Balance of Nature. It's a science book from us, but there's pushback against that concept that nature isn't balanced, that it's dynamic and it's always changing, and maybe we'll have an X year period of lake trout. Okay, we have lots of lake trout now and people are happy fishing, but if we have too many, there's going to be a response, or maybe there's too many, they are not as well nourished because food goes down, and then we have a disease outbreak and the lake trout gets decimated. And then maybe the life rebound. So it's always going to be changing, and it's just a matter of adapting and trying to, fisheries management. I think there's like, let it be, it is what it is, or try to heavily manipulate it, or embrace change. Embrace, okay, well, maybe we're going to get the big head carps in here someday. Well, let's. That's just hypothetical. Crees I popped out. But okay, do we try to eradicate them, or if that's not going to be feasible, do we develop a fishery for them? Do we develop a food market for them? Try to, just adapting to the system.
One more question.
Okay. Yeah.
So Vermont Public this week cited a report from Fairbanks Museum that you haven't been able to walk from Causeway to Law Island since 1934.
Yes.
And that the mean lake level of Lake Champlain hasn't been this low since 1895. I just wonder, are there any opportunistic type of research projects that this drought situation has precipitated in your community there?
Yeah, there's so much, I think. So on the one hand, it's extreme, but like just 2 years ago, we were at record. So this fluctuation is going to be crazy. I think there's opportunities to look at. The lake is so low that we're losing a lot of littoral habitat, so the near shore habitat, and that's habitat for small fish. It's habitat for spawning habitat. Invasive species like tench are shallow, warm water. They're going to be moving out. There's a big biomass of them there. How are they going to affect, like, maybe this new shallow habitat that's in a different zone than will be next spring if it fills back up normally? Then what were the effects in that area when it's usually in this area? So there's just a lot of questions that could be asked for that. Thank you.